Protein-protein interactions. This module will explore the connections that drive biology. This presentation therefore dwells into the essential nature of protein-protein interactions, exploring their types, significance, and diverse applications in biological research and medicine. The module will introduce the topic and then we will look into the methods to study PPI, that is protein-protein interaction. We will also try to understand their role in disease and therapeutics, and finally conclude with the future directions in PPI research. So, for the introduction, understanding protein-protein interactions is fundamental to biology as they form the backbone of most cellular processes. At its core, a protein-protein interaction involves the binding of two or more proteins to form complexes that can exert specific regulatory effects or execute biological functions. They are central to cellular signaling pathways, metabolic pathways, and the overall regulation of cell behavior. Thus, investigating these interactions provides vital insights into how organisms function at a molecular level. Protein-protein interactions. Let's define the core concepts. PPI or protein-protein interactions are the physical contacts between two or more protein molecules forming complexes that carry out biological functions. Its importance in biology are crucial for all cellular functions such as metabolism, signal transduction, and regulatory pathways. Its role in cellular processes include interactions that facilitate cellular coordination and communication, impacting processes like gene expression, apoptosis, and immune responses. Classification of PPI. Protein-protein interactions can be classified based on 1. Affinity and 2. Stability. Based on affinity, they can be classified as obligate. In case one or more of the proteins is unstable in vivo unless interacting and forming a specific protein complex. The non-obligate interactions, that is, in case the proteins can exist independently. Non-obligate interactions can be classified again based on the stability of the complex they form as either permanent or transient. Transient interactions are those that occur temporarily during cellular processes, playing key roles in signal transduction, enzyme inhibition, and dynamic cellular events. On the other hand, we have permanent interactions which involve long-lasting associations that form stable complexes essential for cellular integrity and function, such as in protein subunits of macromolecular assemblies. Another important classification distinguishes the homomeric interactions, where identical proteins bind together, from heteromeric interactions, where different protein types come together. Stable interactions establish permanent multi-protein complexes that can aid in structural assembly, like the construction of cytoskeletal frameworks. Transient interactions are characterized by short-lived connections and are typically crucial for processes that require rapid responses such as hormonal signaling or neuronal communication. Homomeric interactions involve identical proteins binding together, whereas heteromeric interactions entail different proteins forming a complex contributing to functional diversity. How to study protein-protein interactions? So we have a few experimental techniques, all of which aim to identify and characterize the physical interactions between different proteins within a cell. So let's look at each one of them. Yeast-2 hybrid system. This is a method to identify protein interactions by expressing proteins in yeast cells and observing their interactions based on reporter gene expression. The second one is the COIP, co-immunoprecipitation. This is a technique to isolate protein complexes by using antibodies to pull down a specific protein and its interacting partners. The third, protein microarrays, is an advanced technique to screen for protein interactions by immobilizing proteins on a chip and testing their binding to various other proteins. The last one is fluorescence resonance energy transfer, FRET, which is a sensitive molecular technique detecting interactions by measuring energy transfer between fluorescently tagged proteins, allowing visualization in. Next, some of the key computational approaches for studying the protein-protein interactions. One is protein docking. The second one is sequence-based prediction, graph theory analysis, and deep learning methods. All of these computational approaches uses algorithms to predict potential interaction sites and binding interfaces between proteins based on their sequence and structural information. Protein docking is a method that simulates the process of two proteins coming together to form complex by searching for the best possible binding orientations and conformations based on their 3D structures, with tools like Haddock and ClusPro commonly used. The sequence-based prediction utilizes algorithms to identify potential interaction sites based on sequence features like amino acid composition, hydrophobicity, and conserved motifs, often employing machine learning models trained on known protein interactions. Graph theory analysis represents proteins as nodes in a network where edges signify interactions, allowing for analysis of network properties like connectivity, clustering, and pathway identification. The deep learning methods are emerging approaches which leverage deep neural networks to learn complex patterns from large protein datasets, enabling more accurate predictions of protein-protein interactions, particularly when considering structural features. Some important aspects of computational protein-protein interaction analysis. Protein structure database. Utilizing databases like Protein Data Bank to access experimentally determined protein structures for docking and analysis. Interaction databases is the next important aspect to be considered. This includes accessing curated databases of known protein-protein interactions, train prediction models, and evaluate the accuracy of predictions. The third is binding interface predictions. This includes identifying the specific residues on each protein that are involved in the interaction, critical for understanding the molecular basis of binding. Now, why is it important to study PPIs? One is to understand the cellular pathways. By identifying protein-protein interactions, we can gain insights into complex biological pathways and cellular processes. Drug discovery. Understanding PPIs can lead to the development of new drugs that target specific protein interactions involved in disease. Disease research. Studying disrupted protein interactions can help identify the molecular basis of diseases like cancer and neurodegenerative. Significance in biological processes. Cellular signaling pathways. Protein-protein interactions are crucial in cellular signaling pathways, enabling the transduction of signals from the cell surface to internal cellular machinery. Enzyme functions. Enzymatic activities often depend on complex formations through protein-protein interactions, facilitating catalytic processes and regulatory mechanisms. Third is the structural complexes. Formation of structural complexes such as ribosomes and chaperones is driven by protein-protein interactions, essential for maintaining cellular architecture and function. PPIs have been used to construct PPI networks. to study complex pathways for revealing the functions of unknown proteins. So we have the mapping of interconnected relationships with network analysis, biological pathways, and systems biology. These PPIs have been used to construct networks to study complex pathways revealing the functions of unknown proteins. Now let's look at each one of them. Network analysis. System biology approaches permit the visualization and interpretation of complex protein-protein interactions, revealing underlying organizational principles. PPIs are integrated into pathways in biological pathways, which will regulate key biological processes, elucidating mechanisms of action and interaction specificity. In systems biology, which is an interdisciplinary field, it studies complex interactions within biological systems, bridging molecular biology, computational modeling, and bioinformatics. Now, looking at the construction of PPI, we see that it uses mathematical models that consider the structural and functional characteristics of proteins. Next, it uses a variety of effective methods such as shortest path of algorithms, neighborhood-based methods, and diffusion or propagation algorithms. PPI are frequently used to interpret the findings from high throughput studies. Looking at the applications of PPIN, that is the networks, it is used to study complex pathways for revealing the functions of unknown proteins. To prioritize gene candidates in studies investigating the genetic basis of the disease. They are also used to identify disease sub-networks and determine activated pathways in diseased states. Having a look at the therapeutic intervention of protein-protein interactions, we have therapeutic strategies that target PPIs, which are promising avenues for innovative treatments. So, for the beginning, we have small molecules which represent a versatile approach designed to specifically inhibit or enhance PPIs, influencing downstream cellular effects. Two, monoclonal antibodies and their specificity allows for the modulation of interaction pathways that may be contributing to disease. And additionally, peptide inhibitors engineered to correspond to specific interaction domains provide precise intervention capabilities in disrupting pathological network connectivity. We also look at some of the strategies for intervention. Number one are small molecules, that is, low molecular weight compounds that can disrupt detrimental PPIs and offer a novel avenue for pharmaceuticals aimed at specific interactions. Two, monoclonal antibodies, those engineered antibodies that can bind to specific proteins, blocking their interactions and modulating pathway activation. And peptide inhibitors, these are short peptide sequences that can be designed to mimic interaction interfaces, selectively inhibiting unwanted protein interaction. Looking at the implications in disease to understand pathological mechanisms in diseases like cancer, neurodegenerative diseases, and infectious diseases. Altered PPI networks can lead to uncontrolled cellular proliferation and tumor growth, underlining their role as potential cancer biomarkers in cancer. In neurodegenerative diseases, there could be misfolded proteins that can disrupt normal PPI and precipitate conditions such as Alzheimer's and Parkinson's diseases, illustrating their pathological significance. In infectious diseases, protein interactions between host and pathogen can dictate the outcome of infections, presenting novel therapeutic targets. Some significant facts about PPI, protein-protein interactions. Specificity. PPIs are highly specific, meaning that only certain proteins can bind to each other due to the complementary shapes and chemical properties of their interaction sites. 2. Binding sites. Specific regions on a protein called binding sites are responsible for interacting with other proteins. Functional roles. PPIs are crucial for various cellular functions, including signal transduction, cell cycle regulation, DNA repair, metabolism, and protein complex assembly. Transient or stable. Some PPIs are transient, occurring only briefly for a specific function, while others can be more stable and long lasting. And finally, the protein domains. Many proteins contain specific structural domains that act as modular units for protein-protein interactions. And finally, conclusions and future directions. This is to summarize the insights and looking forward. Key takeaways from this presentation include understanding PPIs, which is essential for deciphering cellular functions and potential disease mechanisms, which can inspire novel therapies and emerging technologies. Advancements in molecular imaging and computational modeling are enhancing the exploration of PPIs and their networks. Hence, future research opportunities appear vast, that is, providing potential research avenues, particularly in drug design and understanding of multifaceted protein interactions in health and disease. As we explore the dynamics of protein networks, new therapeutic strategies will likely emerge that harness our understanding of these crucial molecular relationships.